left ventricle. So we have this portion. This portion they cut in this fashion. So this is called a short axis. So you can see in uh, this is the actual MRS scanning. After the short axis scan, it looks like this. This is left ventricle, this is right ventricle, and whatever this portion you are seeing, no, this is called a myocardium. So this portion, this all this are the muscle. So heart muscle. This is called myocardium, and this is and again this is another direction is called fourth chamber. So it covering all the chambers. So left ventricle, right ventricle, left atrium, right atrium, all four chambers are captured in this. And this is two chamber. This is fourth chamber. This is two chamber. Only left atrium and left ventricle. Those are captured in this image. Uh, any doubts in this?
Can you
Okay,
repeat
I
it?
hope.
Flow again, flow. This time I missed it.
Sure, I'll go back.
Thank you.
Yeah. So blood, the deoxygenated blood flows through the okay. First of all, these are called a super vena cava, which are connected to the upper part of the body. This one, and the lower part is called this is inferior vena cava. This is connected to the lower part of the body. When the deoxygenated blood that flows through these super vena cava and inferior vena cava, the blood when flow comes into first it will come to the right atrium. It will go through here and it come through the here. It once it will fill this deoxygenated blood push to the right ventricle. Once this is become it's filled and this is pushed to the pulmonary artery. So these pulmonary artery are connected to the lungs, left side and right side lungs. So what it once it reaches the lungs, again that deoxygenated blood become oxygenated. That oxygenated blood again push it back to the heart through the pulmonary vein. So these are called the pulmonary veins. This portion and this portion. When it push, first it will come to the left atrium. This portion is showing here. Once this chamber fills, that is pushed to the left ventricle through the mitral valve. So basically, this is having valve here. So valve is a main it also, when it fills, that time only this valve will open. So similarly, when this chamber left atrium fills, then this valve will open. Then it will move to the left ventricle. When left ventricle fills, again what it will do, it will push the blood to the aortic wall. So here this is called aortic wall to the aorta. So this portion is called aorta. This aorta connected to the different parts of the body. So this is blood is now this portion is having the oxygenated blood that always is moved is pushed to the outside of the body. So outside of the heart. So it will go to the different parts of the body, brain, like so, like it will go. Any doubts?
Thank you.
Just I'll show one small video here how it pushes how the heart functions. So this portion is left side, left atrium, left ventricle, right atrium, right ventricle, and you can observe here the left ventricle is having this myocardium is having thicker here. The we can say this muscle is more thicker here compared to right ventricle because left ventricle is very important which pushes blood to outside of the body, so it requires more energy here. So that's why this is having very thick here compared to uh, this right ventricle. This portion is very thicker here. uh, any doubts in this? So we can see here all these valves. This is actually a four chamber image that is scanned. Uh, any doubts in this?
No,
No, not from my side.
Okay, sure. Thank you.
No, not from my side.
Yeah, so just we'll go to the application. So this is a advanced visualization workspace icon. Just I'll double click on this. So these are the studies which we got from the hospitals. And all these are the MR modality scans. See if you can see modality, it's showing here MR. Yeah. These are this is a patient name, so this is anonymized, and these are the series which are captured on the different angles, as we saw that 4 chamber, 2 chamber, and short axis. So short axis usually it's a functional series. So if you go to this description, it's showing here something SAWH. This is called short axis. uh, this is it's having more images. So you can see number of images are here 350, and if you click this, you can see here in the quick viewer it will show the short axis. This portion is left ventricle and this is right ventricle. So based on that, just we have to identify which is the short axis. We can select the series which is having more number of images with yeah, something somewhere it will mention ASA something. So based on that we can identify the series. Now, in this ISP, we have different packages like that. We are seeing MR cardiac suite. MR cardiac suite is a package which is having different applications inside that package. So this is the first is MR cardiac suite. The application name is called MR cardiac suite. When we go to review here, if you click on this button, you'll able to see different applications. So only we are concentrating on the MR cardiac suite. So this is the same. This and this both are same. So I'll click this one. Once you click, it loads with the all the images, all the series. Here, whatever we total number, everything will be loaded into this cardiac suite. So this is called a cardiac suite viewer. So here we can see all the images which are captured here. So in this, we can see the short axis. This is what we are looking for the analysis. So radiologists, what they do, sorry, cardiologists, what they will do first, they launch the application data in the cardiac viewer, and they will see what is the so first we go through the all the series which are captured in the different orientations. So we can play here cine. So we can see the heart is functioning. It will play the cine here. And now we want to see this the as I said, the cardiac suite is having different applications. Like if I click here, this is if you click on the new application, this plus assign, it will show the different applications available. So first one is functional LV and RV, means functional left ventricle and right ventricle. So this application studies about the how the left ventricle and right ventricle behaving, whether the blood is pumping correctly or not. So we can study on this. And next comes Q flow. Q flow means this to study about the aortic walls. So if you see this aorta, whether is there any blockage or its blood is moving through this aorta or not. So study about these the diameter or something. Is there any blockage? So study about that. And next comes special enhancement. Special enhancement, it is uh, studying about the myocardium of the heart, whether is there any calcification means is there any problem with the tissue. So some tissues will be died because of blood will not reach to these tissues, and this particular tissue will die. And that time what will happen, the heart will not pump properly. This, so this portion maybe it will become very rigid, so it will not able to push the blood, because this portion, some portion is died. Because of that, blood will not push outside to this valve. So those all things we can study on uh, this special enhancement. And here mapping, this specially mapping is usually this study about the same thing, how the heart behaves when the patient when the patient is having in the stress, means while doing the scan, what they will do, they ask the patient to run on the treadmill. Once the patient treadmill, it will be they give more pressure to the heart, and that time they will check how the heart is behaving, whether it's pumping more blood or it's not pumping, or what is the issue. For that related things, this mapping application is utilized. And next comes temporal enhancement and long axis. Just we go for long axis here. Sorry. Yeah. This also thing just they will. This also thing how the heart is working in this long axis. They want to see on the different angles. For that purpose, they will use this long axis and different temporal enhancement. All these applications. So first we'll go to the functional LVRV, functional left ventricle and right ventricle. Before that, once we load the data, here you can see here label data, and this is option called labeling data. So what it will happen, application will identify what type of these series are. So it will application identify automatically. Once you load this data, it will go and it will check what type series. This is called functional because this is short axis. Always short axis is considered as a functional series. Here scan type. If you see the scan type, it is a functional. And what is the orientation? Short axis. So if you click on this, it will show this particular series. It will show, assuming that sometime application will not identify. It may be something. It will identify something else, something whole art. That time, user have option to go here and select a functional, because cardiologists know that this is a functional series, because short axis by seeing the image, they will identify this is a functional, and they will change this. Suppose if application is not identified, so like that, if you go to 4 channel here, 4 chamber, this also here now it is go to 4. This is showing 4 chamber here, but this is not identified anything. The scan type is not identified the application. That is fine, because usually they're not interested on the 4 chamber sense. This is fine. Otherwise also we can uh, identify the data, what type of data is this. Now we can leave as it is, because this 4 chamber we are not using. Any doubt in this? Any confusion? I'm able to convince you guys.
Yes.
Yeah. Please stop me, okay, if you not understand anything, because these applications are very vast, and the first time going through is a little bit difficult to understand immediately. So you need to hands-on. So at least you have to practice daily. Please stop in between if you have any doubt. Now you
Yeah,
can
sure.
save the. Okay. Now you can. There is a cancel or save option. Once I save this, it will be this will be saved. Now go back to the application. If you select here plus these options, this option come, and I'll select functional LVRV. And here these series are which are related to the LVRV. Those series are added shown in here. Application by itself it will show here, because this is labeled as a functional. Even if you go here, it is this is also considered as a functional in this labeling. So based on this labeling, these applications are identified here. So here, if it is consider, yeah, see here, this is also considered as a functional. This is actual side, but it is considered as a functional. Now, now only we will see. We want to see the only short axis images. So here, if you go and if you see here, it is always if it comes to like this circular shape. This is short axis with the more number of images. So here also you can see here is you mentioned the axial and this is short axis. So always you have to select this one, this series, and click on open. Once you open this application, this data loaded into the LVRV application. If you see, this is LVED, means left ventricle and diastolic, and this left ventricle and systolic. So diastolic ED and ES means when the blood is full, fully entered in the left ventricle. When blood is full in this volume become more here. So if you see, this volume is very higher, and this ES and systolic is, if you see, the blood, even heart pushes blood outside. When this blood moves outside of this become volume, this volume becomes very small. That time you can see here, the this portion is very small now compared to this. So these are the two phases here: LV, ED phase and ES phase. So you can see all this naming convention over here. Any doubt in this?
When you are saying the leveling, it is a DICOM attributes you are talking about, or something else actually you are talking about?
Yes. See, these are the identified based on the DICOM attributes,
Okay.
and there is a algorithm. Uh,
So in each of the series, all the images which are in the series will have this labeling, that is the DICOM attributes. From there only I can figure out this is belongs to which series. Am I right?
Correct, exactly. Based on the DICOM tag, the algorithm, it will trigger on each series and it will identify automatically. And sometimes it may fail. That time, what you have to do manually. Sometimes it's, see, now it's assuming that this is a short, this is not short access, it's a 4-chamber channel, but assume that this is a short access and it is identified as unknown. That time what you have to do, you have to go here, because based on your expertise or experience, rather is what they call is what they'll go and they'll change the scan type. They will make it as a functional. Is that clear?
Yes.
And these are called myocardium. These are the contours. We can see one is red, one is pink. The green, green is called LV endo. This portion, LV endo, and the pink portion called LV epi, left ventricle epicontour, endocontour and epicontour. So these are these are the result application by automatically application generated this when we import the data from the modality. When the data comes to the ISP in this folder, that time application automatically triggers. First it will identify which one is the short access series in this. It will identify, and once you identify the particular short accessibility, on that application will trigger the uh, result. So it will process that particular series and it generates its own results. Once that result generated, here you can see derived series. This is the this series is generated based on this series. This is the original series, and application generated new series, right? FCMR. So I will show you this how it triggers. Assuming that this is a modality, I'm just copying to the some other folder here. Once I copy this, application will trigger automatically trigger the pre-processing. This is called auto pre-processing option feature. So sorry, already this series is already present in this local. So again, it will not trigger now. Already this study is present. We can trigger somewhere else, some other studies. So now I'm copying this study to some other folder. That time application will start triggering. It will start triggering the results here. If you go here, you know, I'm copying this study to SPP folder. There its copying is done now. After completion of this copying studies, automatically it will in progress. It will show the pre-processing. So here you can see the already which the for the other previous studies already this is trigger the pre-processing. So these are the entries related to the pre-processing. So means application generating the results before launching in the into the application. So same register. First it will do the registration between each series. Then based on that, it will trigger the results, FCMR results. So these are the things. I think is that clear or any confusion in this?
What are the things pre-processing does actually?
Yes, pre-processing first actually what it will do, we are given in the automatic automatic option generating the results. So what it will do, it will identify the short access series and it create its own results. Before a cardiologist do any analysis, for them it will give the readily results. It will give what is the present condition of that particular series or that patient. So this is done automatically.
What are these results? What?
Yeah.
Can
Okay.
you-
I'll show you that. Go to application. I'll show you. No, before we analysis, what they want to do, want to see what is the blood in this particular in this left ventricle, how much blood is coming and how much blood is leaving. To identify that, we have to draw these contours. And endocontours and epicontours for this manually. So we instead of adding these manually, application itself identified these contours and generated in this series. So based on these contours, we getting the results here. So uh, this is the auto results. This is application identified based on the present condition of that patient heart of the heart. uh, I think it's confusing, right? It's not clear.
No,
Sorry.
Clear.
it's
It's clear.
Can
Okay.
you just tell me one thing actually? When raw images are first coming, right? And when the
Mhm.
scanning is done, then
Okay.
it will be kept in the packs,
Exactly. Yeah.
and then pre-processing starts.
No. When it will be in the packs, nothing will happen. It will be in the packs only. So there will nothing pre-processing will trigger when it push to the ISP
Okay.
from modality or from ISP. So there are two ways you can push the data from modality or packs. So from whenever it will come to the when it come to the ISP, then only the pre-process will be triggered.
Okay.
Yeah. You suppose if it is in the modality or scanner or in the packs, nothing will be generated.
Okay. After it is coming to the ISP, then only the pre-processing starts.
Yes. ISP has a specific algorithm. It will start. It will identify that series. Then it start pre-processing.
So it's
So-
when you select the data and then click on the application, then it
No,
triggers.
not like that. No. Once it come to this folder, it will start pre-processing.
So the pre-processing is not application specific. It's like a general
Yes.
list of stuff that's done prior to
Ye-
you can open
Yes.
it in the application.
Yes. These are done automatically.
Okay.
Yeah.
So what are
Now,
those common things? That basically it's a common thing, right? Common stuffs will be there for the pre-processing. Am I right?
yes. See, first how it will first it will check the what type of data is coming here. Suppose if I import any other study, it will do the pre-processing, but it will check. It will start to pre-process, but it will not do anything because the required data it will not get. So suppose if that algorithm looking for short access, that algorithm will not get that particular series, then it will not do anything. Just it will leave that is. So nothing will be generated additional here. So this basically this is depends on the number of functional series present here. If this series is present, then only this pre-processing will be triggered, but these new series will be added here. Assuming that this is not there. Assuming that this series is not there, so functional series itself is not there in the scan. When you import this series to the ISP, pre-process will trigger, but it will not generate any results. It will trigger and it will leave like that. If you go here, uh, yeah, see these are the series. It will take each series and try to pre-process these series. And out of that, whichever is valid series now, for that only what it will generate the results and display here in this patient series. Is that clear?
No, it is not clear to me. I don't know why. I'm asking again the
Okay.
question. Imagine
Mhm.
I'm a radiologist and I want to
Okay.
perform one task, and that is someone has given me, okay, MR perfusion operated. Okay.
Mhm.
Now I have done what I'll do. I'll do the scanning. Scanning will give me
Okay.
the raw data. Raw images
Yes.
is come. Raw images are in the packs. Now
Yes.
my objective is what? My objective is MR perfusion or MR diffusion.
Correct.
This is a
Mhm.
application. These are application specific. I want. Now
Yes.
these images are in the raw images are in the packs. Now
Mhm.
some application will input the raw images into the ISP. Once
Mhm.
ISP has come, then my objective is through the to see the images through the MR perfusion or MR diffusion. Now,
Mhm.
now you tell me what is the pre-processing will happen in that case.
Yeah. So those series it will do on preprocess on those series as well, but it will not generate any results unless it will go to each series. It will go this. It will check. It will try to do the pre-process, but nothing will happen. It will leave. Then it will go to next series. It will try to pre-process. Nothing will happen. This will leave, and it will go to next series. So now it's found short access. So on this series, it will generate the results. Pre-processing will triggers on this. Like that, it will go each series. Here you can see that process triggered on each series. You can see 301, 401, 501. But finally, it found here this series. For this series, 501, it has generated the results for this FCMR as well. So like that, it will go with each series. Still, it's confusing.
Let me try to explain to see if I properly understood. When we take data and move it to the local, to the ISP local,
Mhm.
then this process starts. It go
Yes.
one by one each series and see if it entitled to pre-processing. It creates the derived data and put it in the same study in the local
Yes.
ISP local. Is
Exactly.
correct.
Exactly. Yeah. Yeah. Exactly.
Regardless what the system doesn't know what the doctor like to do, just take the data and prepare it.
Yes. Yes. That it'll
Okay.
give it'll give the automatic results.
Okay.
Yeah. So assume that after this, is that clear? Shall we proceed?
Yes, yes, yes.
Okay. Okay. Now assume that you are not happy with this because you are saying that this is not coded properly. Then what you can do, you can click here. This is plus. If you click from the current slice, you can delete the counter from current slice or across. These are different slices here. If you see, total it having 14 slices. All 14 slices it added the results here. So assuming that some slices you feel that okay, this is not according to you. So then you can go and you can just select this option, and from current slice it will delete these contours from this. So this has gone. Now you have option to draw it as a manually. So just to go across, there are two options. One is draw LV endo spline and draw LV endo freehand. So if I go spline, I can just have to select these borders. This is done. Then you can go. Now I'll set, and I have to propagate this because I want to see for other phases also. Just you can click propagate. So this is propagated to here, and once you're done, it will give you the some pop-up. So just you have to dismiss this, and you can see here the this is the results. What is the ejection fraction? What is the stroke volume? The how much ml of blood is moving. So these are the normal values. It's showing it is normal values here. This is for the healthy heart, and this is for the current heart. What is the stats now? For this, it's ejection fraction is 30% only. It's able to push the blood. So, but so required is 59 to 76%. Based on it, will give the different values here. And these are the reference images. So you want to see what is on this slice particular, what is the where on the 4 chamber, where exactly on which slice it is having, we are there. So it will show all these different this one. Here also we can play the cine. You can see here those things. And one more thing is comes automatic segmentation. Suppose I don't want to this again. So again you can application will generate this. If you click on this, other you can first you delete everything. So from all phases and all slices. So it's deleted, entire thing is deleted. So now there is no results now. And I'll click on the automatic segmentation. Again, it will do the segmentation automatically, and it will add the contours. So this process is little bit slower, because again it will do from beginning. It will take all phases and slices, then calculate, and then it will generate the results. So this is a bit slower process as expected. Yeah, again see, again it's generated. And there are different hangings, so we can see different results here. Suppose I want to see the polar maps. So it will give the how much portion is moving it. So this is myocardium, how much the wall, how much this is moving on, right? So if you see here, the wall motion is showing red. So it means there is some issue with this myocardium, because it's not moving properly. So it's in the red color. Maybe some tissues might be these are not functioning properly, it's not moving the myocardium. So that time it will give the results here, red color. So these are different hangings we can go through it. And this is kind of mesh. So how it looks, left ventricle, how it looks. So you can see in the 3D this portion. And here one more thing, there is semi-automatic segmentation option is there. So if I delete this again, I'll delete this. Now I want to do a semi-automatic. Just click here. First I have to identify the wall. So just try to this. Then you have to find the apex. Apex means this portion. This portion, apex. That I have to scroll here. This you can see the small, looks a little bit small volume. Then you have to select this. Then calculate. Again, it will recalculate the contours. It's calculated again, and it giving the results. So basically, all these values depends on the how you draw the contours here. Suppose if I move here, based on it will calculate. It'll calculate and update the volumes. Any doubt here? Okay. So this portion is we are seeing now left ventricle. So here we can see 3 options here. By default, once you launch, it will be in the by default in the left ventricle, because left ventricle is main thing for the relers to study this. Then we can go to right ventricle. If I click right ventricle, it will show the right ventricle only. So this portion. This portion. Now we're studying about this portion. What is the how much blood is coming here, how much blood is going out, for studying of the right ventricle. So you want to see, they just come here. You have to click on this right ventricle, then you can see here. Again, by default it is having only RV endo. So but now you have to draw the RV epicontour. So if you select again this epicontour, just you have to select this spot.
So does this means that part is not part of the pre-processing then?
No, only this portion. Remaining is we have to do it.
Yes.
Yeah, we have to do
Yep.
manually. Yeah. Once you do that, here we can see endovolume. So endovolume, it is showing entire volume. What is the volume right now? You want to see only blood volume, because endovolume means it covers volume of the tissues as well as the blood. But now you want to see only what is the blood volume, only blood calculation. So we have to click on blood volume. So this is water blue portion, this is a blood, and that volume will be displayed here. This is related to the blood only. So this is a right ventricle, we can see in the 3D. And similarly, you want to see both. Now it will display the both. See, this is left ventricle as well as right ventricle. So it's showing result for both here. So now it's the top one show left ventricle, and this is the right ventricle. And it will calculate the what is the heart rate. And these are the BSF. We can change the, we can update these values here based on the height and weight. So again, it will recalculate based on these formulas used here to calculate the what is that volume and all. So any doubts?
No, just one question. Why aren't there any normal values in the right ventricle part? Is that because of the demo, or is that?
No. So this is we are using, this is an old one. It's a RC build. So there is a bug that was resolved in the next version. So this is supposed to be displayed here. That is not showing here. But is a bug.
Okay.
Yeah.
And an old version also outside, you mean, or also only for this demo purpose?
No. It's actually, this is outside only. See here, it's not coming. So this is the issue. We already it's there. So maybe it's in next build it will come. Okay. Any issues? Sorry. Any doubts? Okay. Can I take it as no? Okay. Can we move next application, or you have any doubts in this?
Yes, you can.
Okay. Now we can go to Qflow. These are very small applications. So once we launch application identify, which is a Qflow. So based on this series, you can see the Qflow, and here you first you see IOTA. So this portion you can say IOTA. Just these are some different segmentations provided here. Just do this one as a seed point. Then you can give the different naming here. So I'll give a ascending IOTA. So here also it will show that how much blood is coming from this IOTA. So based on stroke volume, everything same whatever we giving whatever the identifier values, those are displayed in this. Yeah, but again this depends on the how you draw this contour. Any doubt in this? Because you need to just to practice on this, because first time it will be definitely it will be confusing, but once you start hands-on, it will become, you will come to know that exactly how it is. Okay, then we go to special analysis. Yeah.
How the volume through the aorta is calculated? Is the contour change, the contour of the aorta? I don't think the area for the aorta change.
Sorry, I didn't get you. Sorry.
How you here this graph, as I understand, give you the volume of blood passing through the aorta.
Yes, yes. So this is basically, this is some, basically they'll give some contrast enhancement here. So when it contrast is how much contrast
Okay.
coming and how it is contrast going out.
Okay.
So here when it is maximum here, no. So so here
Okay.
maximum
Understood.
blood is maximum blood is coming through this iota.
Okay.
Yeah.
Understood.
Yeah, this special announcement, just the study about the myocardium. So, as I said, is there any issue with the myocardium or any calcification? So that portion is, suppose if any some portion is died, then that will not move properly. So this is identified by application automatically. So you can see this some portion is showing in some other color. So this portion is having some issue. So this portion, so you're having some calcification. So these are some these in this area tissues are died now, so it will not properly push the blood, because it will not move, it will become rigid. So based on this here we can see that, and it is having different algorithm to identify these calcification. It has a different methods. One is myocardium, FWHM, and reference ROI, reference segment. Suppose if I click on the reference ROI, you have to give the tissue which is having very healthier. So just here I feel this is looks healthier, because there is no contrast is standing here. Contrast, everything is moving here, but contrast in this portion it's staying. So there is some issue, because blood is coming but it's not going out on these tissues. So this is because showing here, it's highlighted here. So this looks healthier. So just I'll mention something here as a reference. It compare with healthy with unhealthy. So it will give you the results where and all it is having an issue. So based on that, you'll get the area, the enhanced area. So this is enhanced area, the volume, mass. So similarly, we can see the same thing as a polar maps this year. And assume that it's based on all this expertise or the experience. What they will do, just they feel that okay, this is not good, then they can remove this portion. So based on this, it will calculate what is the enhancement, what is the no reflow, or what is the issue. So it will be identified here. Any doubt in this?
No.
Next we go for mapping. So mapping here, the scanning is done two types. One is T1 native and T1 enhanced. So here native means they'll not give any contrast agent, and enhancements they will give the contrast agent, then they'll do NMR scan. So it will show the mapping of T1 native and T1 enhanced. So I'll select anyone, or either this, or you can select both. No issues. So once you select this, automatically this will be loaded in the application. So I'll click on open. So here it's showing all the short axis images. This one is the native, one is enhanced. So you can see here. Correct. So this is original and this is corrected. So original means suppose you this original as it is scanned done, and this is corrected means application while launching it will do registration, and it will display here. So something maybe while taking the scan, the heart is not constant, it will be moving. So that time, so that is not, sometimes that will not capture correctly. So once you launch this, it will do the registration and display in this corrected manner. It will show here. So if we calculate map, and if we click on segmentation, so it will again it will take the which are result already generated by the application. So here it is showing here. So it is, you can take it on cross one. So again it will show the what is the enhancement here on these applications, on these tissues and all. And these are the values. Slice segmentation wise, it will give the different values here. Okay. Any doubt in this? Again, here same thing. We can do manually. Again, we can delete this and can do the manually. Again, so we can import contour. Suppose this is LVRV. Here, if we do any changes here, and I propagate this, and I want to see these results in the mapping here. That time, if you go here, import contour, this will be import from this application. Contours will be imported to this application. So if I click on this, okay, again these will be deleted, and those contours will be imported to this application in this one, because radiologists have done some changes to the in the LVRV, that they want to see the same results in this application, because this related to the same patient, so they can import the data contours. So again, we can have the different tools. We can move these. Suppose is there anything it's going beyond this myocardium, then we can move this. We can adjust this one. All those these are the simple tools. This one. So any questions on this? Any doubt?
The images which you have shown us, by default, it will be sagittal or axial or coronal.
This is a short axis. This is short axis. Usually, some application for LVRV mapping and special, we need a short axis images. By default, it will based on this labeling, it will identify and it will launch here. As again, so it will not support any other orientation for like 4-chamber or 2-chamber.
Do you have benefit from other than short or long axis?
Because we want to see, suppose how what is the volume of this left ventricle. That time we go for the short. Yeah.
But is there any benefit from other than short axis and long axis, or always we need to convert this?
Always we need to
convert to short axis and long axis in order to start analyzing.
Because see, this application usually the result triggered on the short axis. That's why we prefer short axis. So on any other orientation, the pre-processing will not trigger. That's why we prefer the short axis here.
Okay.
Even we can do that maybe in next version AEW16. They are planning to introduce for the other orientation as well. Now it is only for this trigger. This pre-processing happen out. These results are happen only for the short axis. Any other question? Okay. So temporal, I'm not much aware, but yes, let's okay. Functional, we can go long axis as well. So in this functional long axis, its name itself suggests long axis means it will always you want to see the 4 chambers. So how exactly the all the chambers we can see here in this image. So this is right side and this is left side. This is left ventricle. This is right ventricle. And then also it's a very simple one. And here we can define a segmentation. Just again we can select here. Again, this is ED phase and the ES phase. So based on this 4 chamber, again it will calculate what is ejection fraction, stroke volume, everything calculated based on this contours what we draw. And here no, it will not support this one automatic pre-processed results. That's why it's not showing any automatically generated results here. So we have to do it manually. And here we can see this is a finding dashboard. Here we can see all the results which are application, whatever the application generated results, that will be come and sit in this finding dashboard. You can see this yellow color option. If you click here, it will show all the results here along with the respective polar maps for the applications. Here we can do save this, we can save a different, can see all these results, and we can do the filming and reporting. Those application, these are the options provided here. So any doubt in this?
No.
Okay. So here there is an option called workflow. So whatever we have done, we can create a workflow and we can apply it to the next study. So example, I'll show, I'll delete or close all these. So once you launch data in the viewer, just you go click your workflow and create a new workflow. I'll give it as a test. So my workflow. Then I launch a LBRB. Then I feel okay. Now I feel everything is okay. Then I'll click here. Just it's already new step added, but we can add another extra step here. Add step. We can LBRB and save. So step is saved, and we can launch another application. One go for special. Then okay, after analysis again I will do create one more, add step, save. Now this is saved here, so we can make it as a public. Now it is only for the user specific. Now if I make is mark as a public, it will available all the users of this server. So I'll exit this. While exiting, I'll save these results. I click on yes. Now go back and I'll relaunch this. Okay, now you want to see that particular step you whatever you create, click here test. So it'll remember everything. It's basically it's like a bookmark. So it's loaded here. Then you can see the different steps what we have created here. Either you can navigate or you go and click and select. To a particular step, will step again, and I'll go next navigate. The special will be loaded here. So like that, you can create a workflow and steps on different this one. So any doubts in this workflow? Any doubts in this?
No.
Okay. Yeah, and different we can change the layouts here. So by default, it's launching with 3x4. So if we go here, go to settings here, you can go here and hanging protocols. For this, because this viewer supports different modalities, now we are going only seeing for MR. So you can go here, you can change whatever you want, 16 or whatever is required. Just to click on, or you have to select here 4x4, click OK. So next time onwards, always it will load in this particular 4x4. In this layout, data will be loaded. So the creative workflow can be used for other series or other patients also. The scans. I click this and launch here. So once data loads, I'll I'll go to this particular test, and viewer will be loaded. Then if I go next, the LBRB will be loaded for this patient. So like this. So we can use any workflow creator we can use for any data, any patient. And coming to this here, this one, just a change here. One cross one, and these are the options. So you want to see all these patient name and all based on the option selected here. Show all reference lines, show images, and full image information. If you click on this, it will give all the details of this particular series. So based on that you can use here. And these are the hangings. You want to see particular series. Suppose you want to special rated series, then if you click on this, only the series which are applicable for special enhancement application, those will be displayed here based on the selected hangings. So functional, if you click on functional, all the short access and related series will be displayed here. So you can see all the short access images for this study. Any doubt on this?
No.
Okay, then I have completed this just overview. If you have any doubt questions, please ping me. Yeah, okay. Can we end this session? Or if you have any questions, please we can continue.
I have no questions.
Okay.
Thank you
Yeah.
very much. Very interesting.
Thank you all.
Thank you. Thank you.
Yeah, sure.
you.
If you have any doubts, please ping me.
Okay.
Yeah, thank you all. Thanks a lot.
Thank you.
Bye.
Thank you. Bye-bye.
Bye.
Thank you. Bye-bye.